Today's lecture is going to be fun. We're going to talk about human genome, and we're going to talk about some new sequencing technologies. And I'm going to start with this very historic slide just to show you that it's been only 50 years between when the humans realized that DNA is a double-stranded molecule and its implications of this to actually having the genome sequenced of one of the people who actually described this molecule, right? So it's been a very fast development in molecular biology. And the ability to sequence the genomes gave rise to a new field of science, which we call genomics, right? And the genomics is basically the study of the structure, function, and evolution of genomes. And the reason why we have this new scientific field developing is basically the advances in technology pushes the science into new territories, right? And that technology was the development of the first DNA sequencing machine by Applied Biosystems, which was cheap enough and practical enough for a lot of labs around the world to adapt. So it was based on the Sanger method, which you all know, which is a standard way of very accurate sequencing, and that was 1987. So in 1987, around that time, then scientists started thinking about sequencing the whole human genome, right? And it wasn't actually that obvious to people that should be done. Now, in hindsight, we say, of course, like we should sequence the human genome, but of course, back then, it was more of a debate. But then the decision was made, the money was secured, and the researchers went ahead, right? So the goal was this. Goal was to find a complete sequence of ACTGs in the DNA of an organism. In this case, it was the human. And the challenge was that at that time, there was no machine that takes long one chromosome DNA as an input and give the complete sequence as an output. So the technology was that the Sanger machines could read 500 base pairs or so at a time. So you had to read 100 base pairs, 500 base pairs of a time, the human genome, and then you needed to assemble it, right? So the first group of scientists who took the challenge was an international consortium, which is called the Human Genome Project. It was a public consortium, so the money came from publicly funded, came from public funds, which is basically paid by your taxes, right? So that's National Institute of Health, Department of Energy, etc. It also included other countries and their public funds. It was led by this guy called Francis Collins, who was the head of the National Human Genome Research Institute under NIH. It began in 1990 with a goal of finishing this in 15 years. And the way that they went about doing this was to use genetic and physical mapping approaches and combine it with sequencing to come up with the whole assembly. A few years after Human Genome Project started, a company jumped on ship. This was led by Craig Venter, shown here. It was a genomics company called Celera. It's still going strong. So their goal was to sequence the whole thing with a shotgun approach in 3 years. All right, so what did the Human Genome Project do? So the strategy for Human Genome Project was this. So you start with the genomic DNA, right? So it has all these long chromosomes. And then what you do is you first break up this whole genomic DNA into bacterial artificial chromosomes. And these are about 100 to 300 kb DNA fragments that you propagate in the bacteria as artificial chromosomes, hence bags, right? So, and then once you know which bags to sequence, you divide it into smaller sequences that were about 1 kb or so, and then from each end you sequence, so you put together 500 base pair sequences together with the shotgun sequencing and assembly. So we're going to go through each of these steps one by one. All right, so the first thing is creation of BACs, where basically you randomly divide the genome into smaller length DNA, which is about 300 kb, and you insert it into these bacterial artificial chromosomes. So what the researchers did is they made a BAC library to cover the entire genome. You guys learned about cDNA libraries, etc., right? So the creation of these libraries have exactly the same kind of considerations. So the goal of this BAC library would be that you want to include every genomic region, right? But to do that randomly, you're going to have to make a library that's going to contain some genomic regions over and over again. Right? So there's a math behind this. If you want to take a genome, divide it into 300 kbs, and make sure that your library has every 300 kb, you're going to end up with some 300 kb fragments being there many, many times, right? But you have a certain amount of budget, so you don't want to sequence this whole library. What you need to do is then find the minimum number of BACs that will cover the maximum extent of the genome, right? So that's the next challenge. All right, so you make the BACs, and now you need to find out which BACs to sequence, because you don't want to sequence the same place over and over again and waste your money, right? So the way that they have done this was to use restriction enzyme mapping. So the principle is very simple. So what you do is you take each of your BAC, so here are your BACs, 1, 2, 3, 4, right? And then you compare them pairwise to see how similar the restriction enzyme fragments coming out of these BACs. If two BACs cover the same region of the genome, their restriction fragments are going to be much more similar, right? And if two BACs cover different regions of the genomes, the restriction enzyme pattern that are coming out of those two different DNAs are going to be very different, right? So then you can determine which BACs are covering the same piece of DNA. So what you do is you do pairwise comparison analysis. Okay, so in this example we have 4 BACs, okay? And let's say that since this is random, let's say that these are covering some similar regions of the genome, like here, and then they're also covering different regions of the genome, right? And what you want to do is you don't want to sequence the same thing over and over again. You want to maximize the amount of sequence that you are sequencing with the minimum number of BACs. So what you do is you digest these BACs with a cocktail of restriction enzymes. And because the sequences are different, these enzymes are going to give you different fragments. In this case, let's look at only 2 fragments, okay? Let's say the restriction enzyme A gives you this fragment here in blue. And then let's say your restriction enzyme B gives you this fragment in red. So when you then digest this BAC and run it on an agarose gel, you're going to see multiple restriction enzyme fragments. Okay, so these things are going to contain other restriction enzyme fragments, but we're not going to worry about those. Let's just worry about only 2 of them. And then what you're going to do is, here is your restriction fragment A, here's your restriction fragment B. So one thing that you can say is that restriction fragment B is present in all of your 4 BACs, right? Because you have that DNA fragment here. But your restriction fragment A is present in the 3 of them. So what you can do is then you can compare the restriction enzyme fragments and then order the BACs in their similarity to each other. And you can infer from that if 2 BACs are very similar together, they have the similar number of and the types of restriction enzyme fragments, they're probably covering the same region of the genome, right? So that's a very simple example of this. Does that make sense to everybody? So without knowing the sequence, you use the restriction enzyme fragment sizes to infer if something is similar or not, right? But the human genome is not just 4 BACs, right? There's going to be hundreds of BACs. And the gels look like this. And so somebody, the researchers, needed to go in and compare each one of these BAC patterns to another. So pairwise comparisons. And when they did that, what they could do is align the backs relative to each other, guess where each back is covering into kind of a linear fashion, right? So they said, okay, this back is overlapping with this one because I see these fragments that are overlapping here. This back is overlapping with this one and that one because I see these restriction enzyme fragments overlapping, etc., etc. And then they move down based on the overlaps. Okay, so they're basically trying to put each back in order based on the restriction enzyme similarity. Then they need to find a path. This is what we call the minimal tiling path. That is the minimum number of backs that you can use to cover the entire region. Okay? So for example, if you take this back, then you're covering the sequence here, and then if you take this back, then you're getting the sequence here. If you do that, do you need to sequence this back over here? No, right? Because it's covering exact same sequences, right? So then you skip that one and then pick up the next one that's going to cover another region, etc., etc. So you're going to move linearly down to pick up the backs that are going to give you the most amount of coverage. Any questions? All right. So once you have your minimal tiling path, you're going to have a list of backs in your hand, and you say, I'm going to make smaller fragments from these to sequence, right? So you have your list of backs, and then what you do is you separate each back into smaller fragments separately, right? So you know where each fragment is coming, which back each fragment is coming from, and then you clone these into smaller plasmids, and what you do is you then sequence from both ends. So it essentially, basically you take your back DNA, create about 1,000 bases and put into plasmids. And then if you sequence from both ends, each is going to cover about 500 base pairs, then you'd be sequencing the whole thing, right? Does that make sense? Questions? All right. So the sequencing was done in sequencing centers all over the world. They looked something like this. So there were big rooms with all these sequencing machines, and then these are people who are preparing the DNA. So they sequenced as they prepared, they sequenced and produced all these sequences. Once you have all these sequences, then you need to assemble them. The reason is that although you know a collection of 1 kb fragments are coming from a 300 kb back, you don't know the order of these things, right? So imagine you have a 300 meter rope and you're cutting in 1 meter, okay? They were in some order, but then you jumble them up and then you sequence each of these ropes, let's say, and then you need to put them back together, but you don't know what order they came from. So you need to assemble these things. And for this, you can use shotgun assembly. What do I mean by shotgun assembly? So here's an example. What you're trying to do is you're trying to find out what is the relation of two sequences what's the relation to each other. So what's the order of these two sequences, okay? So let's say that we have a phrase, like English phrase, and we randomly divided that into 4 little pieces. Okay, it's the same idea. You have a 300 kb, you randomly fragmented that into 1 kb fragments, and you sequenced all of them. And let's say we sequenced the phrase, and we came up with these words. Okay, so we have left mid, middle, diddlery, and lurite. Okay, so we're going to now look at 2 fragments pairwise and see how similar they are. So we're going to try and align the sequences, okay? So what we're doing is that we find, oh, okay, fragment 1 and fragment 2 have a region that is very similar, right? So this starts with left mid and the other one is middle. So this MID sequence is overlapping. So I'm going to put them on top of each other, aligning them at this MID, okay? And then what I see is fragment 3 and 4 also have a region that overlap, right? And that's LERE. So I'm going to align these regions and put these two fragments together. And what I'm going to do is I'm going to combine these sequences and say, this is probably what the original fragment sequence was, left, middle, and middle, right, right? And then these two fragments, I'm going to compare them pairwise, and I'm going to realize, okay, the end of this fragment, DDLE, is overlapping with this DDLE, so I'm going to put them, align them together, and then combine them to here. And so my original sequence was left, middle, right. Okay? So in this example, I'm just using letters in an alphabet, but in a sequencing example, you're doing exactly the same kind of shotgun assembly, but you're looking at the similarities between ATGs, the sequence of ATGCs. Okay? Make sense? All right. So how did the human genome cover an entire chromosome? What are the statistics? Okay? Here's an example. So this is the X chromosome. It's 163 megabase. So they used 16 overlapping back clones. So the back clones are shown here. So they're coming from the minimal tiling path, right? The back clones representing, so they had to use 1,400 separate backs. They spanning the whole 163 megabase X chromosome. So the average insert of these backs were 146 kb, and then they divide these back into smaller plasmids and then sequence it. All right. So we call this strategy going from the whole genome into backs, smaller fragments, and then at the end shotgun, the hierarchical sequencing approach, right? So that is the approach that Human Genome Project chose. So they went from hierarchically from the top to the bottom, and when they were analyzing the back sequences, they used markers such as restriction enzyme fragment, right? And they also had genetic markers, which I didn't get into, but they had several markers to locate each fragment relative to each other. Celera did something different. What they did is, I don't care what the original backs and their orders are. I'm going to take the whole genome sequence, randomly fragment it into 1 kb fragments, and then I'm going to let the computer scientists figure out how to shotgun assembly, right? And the problem was that, yes, in theory this should work, but we're going to talk about why it doesn't work. They had to use the original Human Genome Project back clones to actually order their genome assembly. And the interesting thing here is that because this is a public funded project, the data had to be publicly available, right? So the company could go and take the information from the Human Genome Project databases as they produce these things and use the data to assemble the genome. But the other way doesn't work. The Human Genome Project can't take Celera data because it's private. They weren't available, right? So although Celera's strategy worked, the reason why it worked is they used the public data. So the two groups ended their projects and published a draft sequence about 11 years later, after the project started. So these are some of the publications that came out from that. All right. So the end result, one of the things that you might notice is that the original project duration was estimated to be 15 years. It actually ended a lot earlier, and it actually cost a lot less than originally thought because of the technologies that the project developed to make things cheaper and cheaper, right? Okay, so now you have the human genome sequence. What does that mean, right? The sequence doesn't mean anything by itself. What you have is basically a string of A, T, and Gs. And these sequences would fill about 500-page telephone directories. So that's a lot, right? So, and the way that we think of these sequences, we think them as linear, right? So for each long chromosome, we start the sequence from arbitrarily from one end, right? We say, okay, let's decide that this is the left end of chromosome 12, right? And then go one nucleotide at a time across. And the way that you actually present that kind of coordinate, linear coordinate, is like this. So for each sequence, this is a FASTA format. This is a file format that contains human genome sequence. What it says is that it says, here is the information. This sequence is from chromosome 17, starting coordinates from 117 million blah, 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 to 117 million blah, blah, right? So this is the start coordinate, and that's the end coordinate for this little piece. So it's basically linear coordinate. But what we need to know is not just the sequence. We need to know what that sequence means, right? And so that's what we call genome annotation. Annotation means finding where the genes are and the functional elements are, right? So that's what annotation means. So once you have a sequence, how do you annotate a genome? Can you guys guess what you might want to do to annotate a genome? Let's say you have no other experiments. You just have the sequence, and I'm telling you that there is a gene here. How would you look for this gene? You could look for promoter elements, right? We talked about Tata sequences. We talked about initiator sequences. You can scan that little piece and say, are there any sequences that look like a promoter element sequence to me? Then that was going to tell you, okay, there might be a promoter here or not. What else would you look for? Come on, guys. This is like the biggest important, yeah?
Open reading frames.
Open reading frames. Yes, right? So for genes that encode for proteins, finding them is actually super easy. Why is it super easy? Because there are only a certain number of codons that are encoding for proteins, and from years and years of work people have done, we know that code, right? We know that protein code. We know which codon is going to give you which amino acids. And if you have a string of these, you know, okay, I think that there is a protein being made here, right? And with that open reading frames, you can kind of guess where the exons and introns are going to be, right? So that's what the first thing the scientists do. They use computer algorithms based on gene annotations, what we know about the sequence of genes, to find where the genes are, right? And then you can also use experimental approaches to annotate your genome. For example, you can compare the DNA sequence to the mRNA sequences that you would get from sequencing a cDNA library, right? And if you sequence a cDNA library, you will have the sequences of the genomic sequences that encode for mRNA, right? So you would find 5' 3 UTR, you would find 3' UTR, you would find each exon, right? That's coming out of the cDNA. And you can also compare to other species. Often, if a protein or a sequence is important through evolution, right, it's not going to tolerate that many mutations. So the regions of the genome that encode for important proteins like histones or DNA polymerase, etc., haven't changed a lot. So you can find these genes by just comparing, using their homology to other organisms, right? So some of the sequences, features of genes, you can see here. You can look for promoter sequences. You can look for the start codon, right? So that's where the start codon has to start. You can guess where the exons are based on open reading frames. You can use splice site sequences to guess where the introns are, etc., right? And then when you do that, you're going to come up with annotations, which we call gene predictions. So out of the computer models, you're going to predict genes. You don't know exactly where they are, but based on sequences, you can make predictions. You can use mRNA evidence, so the sequence of the cDNA library, you can tell, okay, these are some of the exons in the mRNA. You can use protein evidence, so BLAST, basically homology to other proteins, and then you can combine all these and then come up with some gene models, right? So that's essentially what you see when you open up a genome browser like UCSC. So I know that in your in the recitations you're doing stuff like this, right? So what you're looking at here is in the genome browser. What we're looking at is along the coordinates of the chromosome. So this is chromosome 17 start and then all the way down to 43 million, something, I can't read. And down here, you're going to see all the genes. So they are shown here as little lines, right? And each of these, what is it called? Lines that goes across is an exon, right? And the thin line here is an intron. So that's how we basically express this. And below this, you can find the human mRNA evidence, so the sequences that are also found in mRNAs. You can find where the histone modifications are. For example, here they plot the H3K27 acetylation, and these are mostly found in promoters and enhancers. So based on this, you can say, I think there might be an enhancer right here, right? Something like this. That's another way of annotation, conservation here, and some comparison to other species. All right, so that's what we end up with. We end up with an annotated genome, right? But this still doesn't tell you how the genome functions. It's telling you this is the genome sequences, these are the genes. It's not telling you what each gene does, right? So that said, then the next step has to be finding out how the genome functions. What are the functions of each of these genes? So here's a little summary of what we know. We actually don't know a lot, right? We can predict the function of genes based on looking at the gene sequence and based on what's known in other species. We can guess, right? We can say this is probably a polymerase because it looks like a polymerase sequence. This looks like a histone, etc., etc. And you can do this, and you can assign a function to many of the genes, like shown here. So these are genes involved in nucleic acid binding. So you would find transcription factors here. And what feature of transcription factors would allow you to find them based on sequence? Can anybody tell me? If you looked at a gene sequence, you found the protein sequence, how can you guess this is a transcription factor or not? How do we guess these things? We talked a lot about transcription. Think about transcription factors. Yes? You can do that, but there's so many genes, right? You can do a gel shift with the thousands of proteins, and people are trying to do that, but that's very laborious and very expensive. You want to do this by just computer guess. What do you look for? What are transcription factors? What kind of domains do they have? Activation domain and a DNA binding domain. Within DNA binding domain, what kind of DNA binding domains do we have?
Like
leucine zippers and zinc finger protein sequences, right? And we said that these are conserved, right? And these are similar between transcription factors. They have little differences between amino acids because that determines what sequence they're going to bind. But they're overall, they are similar. So you can look for these similarities. And if you see a zinc finger binding domain in a gene that you're looking at, you can guess that that's a transcription factor, right? And so that's what's being done. So not too bad, right? We know we can guess a lot of these genes, but you can be a pessimist and you can say there's like half of these genes we have no idea what it does. And that's essentially where we are. Even in very well annotated genomes like E. coli, we have a really large proportion of the genes. We actually have no idea what they're doing, right? But they're functional because they're conserved. They're there, right? So they must be doing something. So then what should we do to determine function of all of these genes? What kind of research do we need to do? Can you do research on humans? Can I just knock out one of your genes and find out what happens? What do we need to use? Model organisms, right? So we use genetics. We knock out genes, find out what happens, right? Or we do other tricks to find out which genes are doing which functions. And we do these in worms and flies and fish and stuff like that, not necessarily humans, right? All right. And the reason why we do these is because we are descendants from the same. We're descending from the same common organism, right? So we have a lot in common with microbes in C. elegans, Drosophila, Arabidopsis, mouse, and humans, of course, because we share a high percentage of genes in common. And some of these genes are involved in diseases, right? And that's good because then we can study these disease-linked genes in model organisms and then model it for humans and guess what they do in human diseases, right? So essentially, because of this, and sequencing the human genome dramatically increased the rate at which genes are underlying Mendelian disorders, right? We found a lot of these single gene type of diseases. So these diseases caused by mutation of single genes, they're easy to find. The complex genetic diseases remain very challenging. Give me a complex genetic disease. Obesity, heart attack, anything. Cancer, we could say that, right? A lot of these things that are killing us now are basically complex diseases, right? The single gene ones are affecting a very small population, but small number of individuals in our population. All right, so what came out of this human genome project? A lot. We have made a lot of progress in a lot of fronts in this, and some of this was surprise. Some of the findings of the human genome was actually surprise to even the scientists. Right? So before we knew the genome sequence, some of the papers and reviewers wrote that they predicted about 100,000 genes in the human genome. And the way that they based this is before the human genome was sequenced, the researchers practiced on smaller organisms' genomes. So they sequenced yeast, and they found about 6,000 genes. They sequenced C. elegans. They found about 20,000 genes. And they said, okay, yeast is a simpler organism. It has fewer genes. Worm is more complex. It has more genes. Humans are more complex than C. elegans, so we should have more genes. But it turns out, no. We have just about the same number of genes as a worm, right? And Drosophila. So where do you think the complexity comes from? Alternative splicing. Worms have alternative splicing too.
Regulation.
Gene regulation. That's where the complexity comes from. Think of genes like Lego blocks, okay? So you have the same types and same types of Lego blocks, but it depends on how you combine them, right? So all that complexity comes from gene regulation. So it's not enough to just know what the genes are. It's not enough to just know what the Lego blocks are. We need to know how they're put together, right? And the other thing that came out, which is not a surprise to a lot of people who understand that evolution is the basis of biology, is that there are large regions of our genome synthetic with other organisms, right? So we had large chromosomal regions that are completely synthetic with mice, which looks very different from us, right? But that says that at some point we came from an ancestor that had the same chromosomes. All right. So when the sequences from mouse and humans were compared, we find genes that are in the same order along long stretches of chromosomes. That's what synteny means. You find the genes in the same order along the chromosome. So if you take all the genes in the human genome on chromosome 17 and align it with some other chromosome 11 in mouse, what you're going to see is a lot of the genes are in the same order along the chromosome, right? So which highlight actually took advantage of this kind of synteny? We talked about in the past few lectures, we talked about a study. They use this kind of synteny to study what human disease. Cancers use this a lot, but it wasn't actually cancer. Fingers. Remember how the chromosome deletions and translocations resulted in brachydactyly? And if you want to study that, you can model it in mice because the genes that were involved in this were in the same order along the chromosome, and the chromosome's topology, like how the genes were, how the enhancers were activating genes, was conserved, right? So you could make the same deletions that you see in humans and produce the phenotypes in mouse, right? Amazing. All right. So far, we're all content, right? Human genome is sequenced. Everybody happy with this? Fine. But whose genome was it? When we say human, who do we mean? Right? When you sequence somebody's genome, one DNA, do I represent the all the humans on Earth? No, right? So that human sequence is just coming from a few humans. Whose genome, right? Whose genome are we talking about when we say human? When we're talking about when the human sequence and show those UCSC browsers, we're talking about something called a reference genome. So that's the starting point. That's the starting sequence that is isolated from a few individuals giving us that sequence. But that doesn't mean that's the only sequence that the humans have, right? So what about the two projects? Whose genomes did they sequence, right? So both groups basically collected blood and sperm samples from anonymous male and female donors of different ethnic backgrounds. Sperm is good because you can make high-quality DNA. That's why they were collecting that. And then they sequenced it. So the Human Genome Consortium said that we do not want the resulting human genome reference sequence to represent only one individual. Okay, so they said, okay, let's take from 10 to 20 different individuals across different racial and ethnic groups to try and mix up, right? So that was public. What did the private do? So the private also had the same idea. So they had 5 individuals here. But if you actually look at the number of sequences that came from each of the individuals, what you're going to find out is that one individual is contributing most of the DNA sequence. And guess who that was? Who is a narcissist? So that was the company owner, right? So basically he sequenced his own genome. All right, so nowadays we can actually sequence the genomes, the whole genomes of individual people. So it started with Craig Venter when he donated most of the sequences in the Celera sequencing project. Then the Watson's genome was sequenced in 2007 for $1.5 million. What a waste, right? So it took about 4 months. So the initial human genome took many years, but then because the sequence was sequencing technologies developed, you could use next-gen sequencing, etc. And then Steve Jobs got his genome sequenced, but he didn't believe in medicine, so that was too bad for him. It was about 100K. And now sorry guys, am I like totally incorrect? All right. All right. So in 2012 and beyond, it cost about $1,000 per genome to sequencing, and because the reason why the cost went down is then the technologies have developed a lot and they're cheaper and cheaper. Okay, but we actually just don't want to have these rich guys' genomes, right? I'm interested in how they got rich, but I'm not that interested in how they got bald. So what we need to understand the human genome is that you need to sample many, many genomes, right? So when we say human genome, we need to have a holistic understanding of what this sequence means. So there are new projects, new human genome projects, and one of them is called 1,000 genomes, right? So this 1,000 genomes consortium said that, okay, let's catalog the human sequences from different humans so that we can understand the variation and we can understand what the human genome means, right? So as of a few years ago, they had 2,500 or so individuals sequenced across Africa, East and South Asia, etc. And that number is actually so much more than a few thousands now. I don't actually have that number because now we can, right? We can sequence many, many different people. All right. So one of the difference between the sequencing of these new genomes and the original reference human genome is that these genomes are not assembled de novo. What that means is that they use the reference genome to align the sequences. Okay, so that alignment is actually the biggest problem in determining sequences de novo. De novo means like new, like without knowing anything. Okay, but these ones now, what we do is we align the sequences on that scaffold of the known reference genome. All right, so what does that mean? So in the original Human Genome Project, the order of the sequences were determined to create this reference sequence where we have an idea what these sequences are. Now, when we generate a new person's genome sequence, what we do is we isolate the genomic DNA, break it into random pieces. We generate millions of sequence reads, and then do not shotgun assembly. And we take all these reads and then align it to the reference sequence, and then we to try to understand what nucleotides are different between the reference sequence and the sequence of the genome that we sequenced, right? So on average, between us, about 0.1% of our genomes are different. So out of 3 billion positions, that means about 3 million sites, we are different from each other, right? All right, so far we're again comfortable. It's fine. We have the sequences of a few individuals, and we're collecting more and more sequences, but there is a problem. The problem is this. Although we say the human genome sequence is complete, it's not entirely complete, okay? And the problem is this. In 2004, the Human Genome Project reported 341 gaps in the genome. In 2017, the number of these gaps haven't reduced that much, okay? We still have these gaps. The reason for these gaps is that it's very difficult to determine the sequence of the repeat regions of the genome. And what percentage of our genome are repeats? About 50%, right? It's a pretty large percentage of our genome. So for example, our centromeres are not fully sequenced because they are long stretches of DNA satellites, okay? The reason why assembling repeat sequences are very difficult is that repeat sequences directly interfere with shotgun assembly strategy, okay? So let's go back to our original example. So let's say that, that our phrase now is left, middle, right, but I put a little repeat sequence here, which is LE, okay? So this is representing the repeats in the genome. So we have a LE, 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 okay? So we generate these fragments again, and we try to pairwise compare them, but every time we pairwise compare them, what we're going to have is we're going to have these repeats aligning to each other, okay? And because there are many repeats, you're not going to know, for example, this LE is aligning here, creating this left middle, or it's aligning the other way, in which this LE is aligning with here, and that will create a completely different sequence, right? So that's a problem. So in principle, a genome of any arbitrary size can be directly sequenced by the shotgun method, provided that it contains no repeat sequence. And that's absolutely not true, right? We have a lot of repeat sequences in a lot of animals. So it's very difficult to just use shotgun method to assemble. Does that mean we should give up? No. What we can do is we can do the best we can do, right? So if you want to take a new animal and you want to sequence its genome, this is what you typically do. You take your sequence genome, the genome of your interest, you divide it into small pieces, you make these random DNA fragments, and then you sequence each of these random fragments, And then you use shotgun sequencing, right? So you're looking at the overlap between sequences to create these contigs. Contigs is basically contiguous segment assembled from overlapping sequence reads. These contigs, you can't create the whole chromosome because of the ambiguity that these repeat sequences give you, right? So you're going to get as long of a contig as you can by using shotgun sequencing. And if you can order these contigs, you're going to get scaffolds. That means the ordered contigs without any gap. So if you don't have any gaps for the whole chromosome, then your scaffold would be the whole chromosome. If you have gaps like centromere, right? So your scaffold is going to be, you're going to have two scaffolds from the left end of the chromosome and the right end of the chromosome with the centromere, which is the gap in the middle. Does that make sense? And so whenever you hear in the news the new genome sequence of this blah blah was sequenced by high throughput sequence, this is probably what they've done. Okay, and a lot of organisms and crops of economic importance are, for example, sequenced this way, so that we can actually know the DNA sequences. All right, and the sequencing technology allows us to sequence more and more organisms, so we're not only sequencing humans, we're sequencing viruses and prokaryotes and eukaryotes, and so the number of genome sequences in the public databases are increasing with time, right? All right, so here's, for example, one organism that was sequenced. It's Atlantic cod, right? So when you look at the sequence statistics that they have provided, now you should be pretty good at reading this, right? So it says that the number of contigs is this many. The average number of bases in each contig is this much. The number of scaffolds that they could assemble from these contigs is that many. The entire assembly has this many number of bases, right? And then so you know that, okay, this is the genome assembly, but the whole genome is divided into 6,400 scaffolds. So we don't, if they have, I don't remember how many chromosomes they have, but depending on how many chromosomes they have, you kind of have an idea of how fragmented the genome is. So the problem is then having these gaps, right? So we can only get as good as scaffolds. What kind of sequencing technology do we need? What do we need to do to fill in these gaps? If you were a technology developer, what would you want to develop? What kind of technology do you need to develop? Ideas? Yeah?
Something that deals with recombination.
That's something that what?
Something that deals with recombination.
Deals with recombination, deals with repeat sequences. Right? Something that will deal with repeat sequences.
If you can like sequence a longer part, then like you know more about like where the repeats are.
Exactly. So recombination idea, you're going to have to elaborate that to me later, but that's the idea what people are going for. They're saying, let's develop technologies that's going to take out one end of the DNA and then go all the way to the other end. That's going to solve your repeat problem because it's going to tell you exactly what the sequence is, right? Very simplistic, but that's kind of the idea. And there are now technologies that are actually being developed, and one of them was this really new paper, and very exciting paper that could linearly assemble a human Y chromosome, human Y centromere using nanopore long reads. So they were capable of reading the Y chromosome centromere from beginning to end using this new technology, which we're going to talk a little bit about. The reason why they used Y chromosome is the centromere is smaller. That's why. Okay, so they're starting easy. Okay, so technology. Technologies are important in this business, right? Because it increases our capabilities. And so you're going to hear this next generation sequencing technology. Next gen sequencing basically refers to everything after the Sanger sequencing. So this next generation sequencing, some of the leading companies are Illumina, which you learned about, 454 Solid, Helicos, and PacBio. They differ in their strategies for how they prepare and detect the DNA sequence. Okay, so we learned about the Illumina sequencing. And in Illumina sequencing, what we have is that we have the DNA that is attached to the flow cell, right? And then what you have is you have your each nucleotide that is conjugated to a different fluorescent dye. And as the polymerase adds each of these fluorescent nucleotides, the fluorescent molecule gets cleaved when the nucleotide is added. And because the scanner is scanning which nucleotide is added at each DNA spot, it's going to tell you which nucleotide it is added from which color it sees, right? So that's basically the idea of the Illumina sequencing. The polymerase is adding nucleotides, and every time each nucleotide is added, you have a scanner telling you what nucleotide is added. There are other strategies, such as ion torrent. So in this case, what you have is you have your DNA that's attached to a bead, and there is a semiconductor that measures the change in charge as each base is incorporated. So as each base is incorporated by the polymerase, a single H ion is released, and which is detected by the sensor. Okay. So what they do is, instead of putting all 4 nucleotides at a time, they add it one at a time. Okay. So whenever they add the A nucleotide, if your bead contains the DNA sequence where there's a T as the next nucleotide, right? It's going to add the A, and so if you see a signal from your detector, you're going to say, at the cycle, when I added the A nucleotides, this got incorporated into this bead, right? So I know that that nucleotide in the template was a T. So when you add another A in the next cycle, let's say, is it going to get incorporated here? No, right? So you know that your next nucleotide was not a T, right? And then when you add a T in one of these cycles, it's going to get added, and you say, okay, in this molecule, in this bead, the next sequence was A, right? So with that, every time you add and you get a signal, you're going to have the sequence of your template. And that's essentially what this technology does. Any questions? All right. So in both Illumina and in a lot of technologies, what we have is we have the DNA polymerase, right? That's freely going in to the template and starts adding nucleotides. So that's how Sanger sequencing is done, how Illumina is done. In this next technology called PacBio, that PacBio company uses a lot, what they do is they don't have a free DNA polymerase. Instead of attaching the DNA to the surface of detection, they attach the polymerase to the surface of detection. Okay, so what they do is they immobilize this polymerase that's shown in green here on this little plate that is a detector. So what are then the DNA is here, in the template DNA is shown in gray, and your growing chain that's the nucleotides are added by this DNA polymerase is shown in orange. Okay, so this is called single molecule sequencing because the detector detects the addition of nucleotides at each polymerase, at each site. So it's sequencing the single molecule that's attached to this polymerase. So how does that happen? So what's happening at the surface of the detector is this. So you have the detector here, you have the immobilized DNA polymerase here, you have your template shown in gray, and your growing chain is shown in purple. So what you do is, in your reaction, you have 4 different nucleotides, and each of them is coupled to a different fluorophore, and every time the polymerase adds this fluorophore, the chemically bonded fluorophore is going to be released, okay? So what you have is you put your nucleotide in. If the polymerase catches this nucleotide and attaches to the growing chain, there is going to be a short amount of time in which the fluorophore is going to be very close to the detection surface, okay? So the detection surface is going to then record the color of this fluorophore in a fluorescence pulse. And after this nucleotide is added, this is going to be cleaved, and it's going to move away from the surface, washed away, okay? So when your next nucleotide comes in again, the polymerase, when it's incorporating it, there's going to be, again, a little delay in which the fluorophore is going to be very close to the surface, and the detector is going to read it, and then it's going to be released, etc. Right? So it's going to go one nucleotide at a time, reading this single molecule sequencing. Make sense? All right. So here's another technology. So far, everything depended on polymerase, right? All of those technologies we talked about depended on the polymerase, adding a nucleotide that's complementary to a template. In this technique, we don't have that. What we're doing is we're reading the shape of the DNA. And the way that that's done is by sequencing the DNA, by threading it through a microscopic pore in a membrane. So the bases, each base is identified by the way they affect the ions flowing through the pore from one side of the membrane to the other. So the charge and the chemistry of each nucleotide is going to, each base, ATGC, is different. And the way that they are going to affect this ion flow that's going through this pore is going to be different. And that's what the detector is going to read, okay? So what you have here is you have a membrane where you have a protein that's fixed into the membrane, and this is going to push the DNA in here. So the DNA double helix needs to be first opened up by a protein that unzips the DNA helix. What's this protein? Helicase, right? So it has to be a helicase. And a second protein basically creates this pore in the membrane and holds the adapter molecules basically here. The adapter molecule is holding the DNA, keeps the bases in long enough for them to be identified electronically, and this thing is going to, as it opens, push the DNA in. All right, so every time the DNA is going in, it's getting pulled one nucleotide at a time, okay? And that each nucleotide at the site of the detection is going to disrupt the flow of ions in a stereotypical way. So based on that way, which you're going to read here, you can determine what nucleotide was at that site at the time of the detection, right? So you're going to read one nucleotide at a time based on the property of ATGC bases going through. All right. So these are super exciting because PacBio and Helicos, for example, can read very long DNA fragments. These DNA fragments can be in the order of tens of kbs, and people are pushing it to go even further, right? So you can actually read one thing from beginning to end. But just like everything, these are not perfect, right? Life is not perfect. So each of these technologies have their pros and cons. So here is a table that's summarizing some of the problems. So Illumina sequencing is great, but the problem is that it can give you only short reads. And these are not very useful when you're trying to do shotgun sequencing of genomes containing lots of repeats, right? The good thing about these is that they have a very low error rate. That means when they read the sequence, they give you the precision of that reading is very good. They don't make very much error, right? So that's good. The other technologies, such as the nanopore technology, that like the moving of the DNA through that little pore, gives you really long reads. You can go up to 200 kb. But they give you a lot of errors during reading, right? And that's not good because you want to read the whole thing, but you don't want to read it just with lots of errors because that's not what the information will be wrong, right? But because now we have a mix of technologies that can read long versus they can do short reads in very high accuracy, you can combine them, right? So what you can do is, if you have a new genome, Sanger sequencing with back method is super expensive. It's gold standard. It's just expensive. So we don't do that. What you can do is you can choose two cheaper technologies. So based on what I just tell you, for example, what two technologies would you combine to sequence whatever organism that you want to sequence?
Use the nanopore to put Illumina sequences in order.
Perfect. You can use the nanopore, that long reading, to determine the order of the sequences, right? But the actual identity of the sequences, you would base it on the Illumina short reads. And that's essentially what the researchers are doing right now, which is with the availability of these techniques, most of these problems are becoming an engineering problem, right? So for example, this is, let's say you have one technology. The problem is that it's not very accurate. How do you increase accuracy? The way that you can increase accuracy is you can read the same thing over and over again. That's what that means, is you increase the coverage. You read the same region over and over again, and in each read, it's going to make one random mistake, but you can use that randomness to your advantage of assembling the sequence. For example, this is your first read. This is your reference genome. You can have multiple reads, right? What your engineering goal is to optimize the number of reads that you need and the cost to have the maximum accuracy of the reference genome. So what you can say is, for example, if you cover the same region of the genome over and over again, and in most of your reads, you're going to see an A at this point, but in a small fraction of your reads, because the error is random, you're going to see a C, right? Then what would you decide your reference genome is? You're going to decide it's an A, right? And likewise here, if most of your reads have a G, but at some point in some of the reads you have a C, you're going to decide that this is a G. All of these engineering problems require statistics, okay? So here's something I'll tell you. If you haven't taken any statistic courses, definitely take it. In anything that you will do in your life, which basically is going to require data and is going to require data analysis, you're going to need statistics, okay? That's the most important thing you could probably take out of your college. Go take a statistics class, because these decisions are made based on statistics, right? Deciding how many reads do I need to significantly say this is the right nucleotide or that's the right nucleotide. All right, where do we use these technologies? So it's not just in the lab. You can take these things to the field, and so there are a lot of applications of this because these technologies are now pushing in that the sequencing instruments are getting smaller and smaller. So this is a handheld sequencer. So here's the researchers putting the DNA right there in the sequencer, and this laptop is going to basically read. And for example, people are trying to use these to fast identify a pathogen in the field when there's an outbreak of a certain virus, etc., right? And now the sequencers are also taken to the space, and where this astronaut / scientist basically can sequence the DNA in a space, which is amazing. Yeah. All right. So that's it, and I'm gonna see you guys on Wednesday.